Thank you, Dr. Lin. So it's a pleasure to introduce Dr. Evan Yu, who's a professor of medicine at the University of Washington. He's a medical oncologist and has had many years of interest in developing and utilizing better approaches to image prostate cancer, primarily using PET scanning. So we'll hear about that. Evan?
Thanks, Pete, and I want to thank you all for being here today and listening to me talk on a Saturday, a rainy Saturday, that being said and done. But I just want to clarify one thing. My talk is focused on PET imaging and finding the cancer, finding local recurrences, but I tried to leave the fancy words out a little bit. So the other component of it, as you read in the program, is about oligometastatic disease. I figure I'd just define that a little bit since I left it off the title. Basically, oligo means a single, and metastatic means that it is spread outside the organ and started with. So the idea that if you find something like a single site where the cancer is spread to, or sometimes we expand the term oligo to mean more than one, that perhaps we can do something about it that might actually lead to really good outcomes, and we'll be talking about that in this talk. So there's a lot of reasons to image, and I think many of you know that we do things like CAT scans and bone scans for prostate cancer, but there are certainly fancier imaging modalities, as Dr. Lin just showed you there with all the cool MRI imaging they're doing. But first we have to think about why would we image somebody, and of course there's a lot of reasons. Screening, Ruth talked a lot about PSA screening, but there have been studies with imaging for screening, certainly more expensive modalities for screening, but that's one reason. To make the diagnosis, to find the cancer initially, and initial staging. Once you've made a diagnosis, how extensive is that cancer? And then restaging for recurrence. That means ideally somebody who maybe had prostate cancer, had surgery, had radiation, and then unfortunately the PSA comes back up and we think the disease is back. Can we find that cancer? Can we do something about it? And of course, something that's been an interest of mine, as Pete's alluded to, is response to therapy. In the past, we've done quite a bit of PET imaging research here with novel PET imaging modalities to look for response to therapy. Who's doing well with the treatment? Who's not doing well with the treatment? So we can change treatment early if needed to really try to get the optimal outcomes. But today, what I'm going to focus in on is more finding the cancer and what to do about that. So initial staging and restaging for recurrence. So let's ask the question, why is it important to find the cancer? What are the real reasons to do more fancy imaging that might detect the cancer at a lower PSA level? Well, there's a couple main reasons. Number one, which is what we're all hopeful for, is can we find the cancer early, and can we try to treat that potential oligometastatic or one-site lesion that is spread and cure the patient, obviously. And if we can't cure, maybe we can do another fancy word, cytoreduce, which is reduce the tumor burden so that we lead to still better outcomes down the road. Whether we do that with surgery, radiation, some new modalities, the whole idea is can we find it and can we go after it, okay? But there is another reason as well, and that is if it has disseminated, spread widely, do we want to go after it? Okay, we have to think about this because unfortunately there is no free lunch in life, and with everything you do, there are potential downsides to it. So if you just can't cure the cancer, we do have good life-prolonging therapies out there, but do you want to go up and do surgery? Do you want to do radiation? Do you want to do treatments that might lead to some side effects? Of course, Dr. Lin's surgery never has any side effects, of course, but minor side effects, honestly speaking. But that being said and done, we have to think about this. We have to be mindful because we're in the business of helping people here, right? We're not in the business of giving side effects, so we're always trying to figure out what the best thing to do is for each individual patient. All right. So what is PET imaging? Well, we mostly combine PET imaging with CT imaging, and I think the way to think about it is CT imaging is like taking a picture, okay? You're taking a picture, what does it look like? But PET imaging is looking at the activity of the tumor, okay? And the nice thing about it is we can quantify. We can give it a score, a number. Is this more active? Is this less active? And we can look at a lot of things. We can look at metabolic activity. Is this thing taking up a lot of glucose? Is it, are the cells building up? And around every cell there's a lipid membrane, so we can image lipid metabolism as well. And then we can also do molecular imaging. Can we look at imaging just a particular protein that's expressed on that cancer cell? And so that is really PET imaging, is you're combining a picture, anatomic imaging that gives you a picture with activity of the tumor, okay? And it could be advantageous compared to things we do in this day and age. For instance, PSA, that's kind of a summation of what's going on. You add together PSA being produced by the cancer, you draw a tube of blood, it's simple, it's cheap, it's quick and easy. That's great, but at the end of the day, it's a summation. And there is heterogeneity in cancer. There's another fancy word again, but heterogeneity just means that not every single lesion in your body may be created the same. And we talk about genetics of cancer, it can change, it can mutate over time, unfortunately. And so one spot may not be equal to another spot. They may behave differently, they may look differently, they may act differently. And so that's important. That PSA is just a broad summary. Okay, it doesn't give you information about every single spot. Now, that lays the groundwork for if you biopsy a spot, which we're doing a lot of research with here to try to understand the genetics of the cancer, but if you biopsy a spot, we have to recognize that may not give you information on every single spot that it's spread to, whereas PET imaging may give you information on every single spot, and we sometimes see this where a lesion gets less bright or a lesion gets more bright in the patient. There's mixed responses to therapy where some are getting better, others are getting worse. So it's important to keep in mind the advantages of why we do this. So let's talk about PET imaging in prostate cancer. Now, the standard PET imaging that people have done for cancers, lung cancer, lymphoma, all different types of cancers, has been FDG PET imaging based. And that is kind of like, looks like glucose, molecularly like glucose. So it's uptaken in rapidly dividing cells. That works in some prostate cancer patients, but it's not ideal, okay? It hasn't worked real well. So we've looked at other modalities here, and we looked at acetate imaging here in particular, which is a method of lipid metabolism, similar to choline as well. And choline did get FDA approved, but it was not widely available and only available in really one center for the longest point in time. But I want to lay the groundwork because this is not the latest and greatest anymore, but for a while it was, where choline could be used to try to find the cancer locally. It has it spread to a lymph node, okay? So this is a study where basically they took patients who had surgery, radical prostatectomy, and then their PSA went up. That's what biochemical recurrence means. And they looked to see if they did imaging with choline PET, could they find the cancer? And the reason I highlight this area is what it points towards the fact is that you really need to have a PSA of around 1.5, okay, to find that lymph node, to find that cancer. Now, the challenges are in this day and age is after surgery, and if you have your PSA go back up, we've learned that if you do something about it sooner, if you're going to do radiation, it's probably better to do it at a lower PSA, for instance, 0.5 or less. And so 1.5, if it takes a PSA up to 1.5 before you can detect it in about half the patients, maybe that's too late already, okay? So that being said and done, you know, we've kind of moved on. So here is a new modality of imaging called FACBC, flucyclovine. Another term for it is Axumin, A-X-U-M-I-N. Okay, that's the kind of FDA-approved pharmaceutical name for it. But this is just basically an artificial, unnatural amino acid. So you might say, well, what is that? Okay, well, it makes up protein. And for cancer, similar to lipid metabolism, how it has a cell membrane around it, glucose metabolism, how it needs to uptake glucose to grow and survive. But proteins, obviously for a cancer cell to grow and survive and spread, it also, you know, has to synthesize or make proteins as well. So this is actually an FDA-approved modality. And as you can see here, FACBC seems to be able to pick up prostate cancer at low PSA levels, for instance, 0.5. And there's an older imaging study, which I'll talk a little bit more about, Prostate. It's FDA-approved, but we don't really use it much anymore. But as you can see, you can pick up this cancer with FACBC earlier. So the way to think about these images, these CT images, are really your head is laying in the screen, your legs are coming out, and this is a cross-section image, okay? So really, this is the left side of your body, and that's the right side of your body, okay? And so you're just taking cross-section pictures. But you can see that the cancer pops up here, okay, with the FACBC imaging at a very low PSA level. And we can image the cancer outside of the prostate as well. And you can see it just doesn't pop up on prostatin. So I'm showing you some pictures here of what FACBC imaging looks like. But the important thing to know is, can we do it here yet? And we will be able to soon. I don't show this slide to really go through this in detail, but to go through the fact that unfortunately there's a long regulatory process to do imaging studies in this country. And this is how it happened with FACBC. It went through a long process, and it's licensed to Blue Earth Diagnostics now, which is a small company, and they have not rubbed up production facilities yet in the Northwest United States. Okay, so it's coming soon, probably end of the year or next year, within the year. Hopefully we'll be able to do this, but this is coming, and that's why I wanted to share it with you, because it's something to keep an eye out for. Okay, now there are other things coming that are earlier in development than FACBC. There's something called PSMA imaging, and PSMA is prostate-specific membrane antigen. Okay, this is expressed maybe on about 98% of prostate cancer cells in the prostate. If it's spread to other sites, there have been some studies that show it's about 94%, but pretty much most prostate cancer, whether it's in the prostate or spread, expresses this protein, okay? And this picture here is interesting because it shows that, remember I talked about processing, how it's not great? When we see the cancer there, it's only about cancer half the time, and about half the time you have cancer there and you don't see the cancer there, so that's not a great imaging modality. Well, this image is a different part of the protein. This image is the outside, the external binding part of the protein, whereas this image is the internal component. So that's one major difference, and all the PSMA imaging modalities, because there's more than one, that I'll talk about image to outside. Okay, so this is an antibody that images outside, and as you can see, it picks up more than what a bone scan does. These are older images. This is already like, almost 15 years old, but these are the early attempts. What I'm going to show you subsequently is better. But I think the thing to bring up is that all the new imaging modalities image the external part of the protein here, and so gallium PSMA is a radio-labeled form of detecting PSMA that is much prettier, as you can see with these images here. And when you compare it to choline, because remember I told you choline is approved, but you need about a PSA 1.5 to find it, this can find it at a much lower level, and this just shows how much more delineation of the tumor that it has here compared to choline. So it's just much better quality images and much greater ability to detect at lower PSA levels. So this graph gives you an idea of just how low of a PSA you can go to detect it. Here, as you can see, 0.5 range to 1, 72% had detected by this modality. If you want to talk about 50%, well, here at 57% detection at 0.2 to less than 0.5. So at very low levels, you can start to pick this up with gallium PSMA. Now I show this imaging slide to show that there's different forms of PSMA imaging. PSMA is the protein target. The gallium component is the radio-labeled compound that allows it to fluoresce, to show up on the imaging. Now F18 is what we've standardly used, like for instance with the glucose metabolism imaging, FDG is F18 as well. The advantage to this is that it's easy to transport. F18 has a long half-life. But this is being done as well. I'm not going to get into which one's better, which one's worse. There's a pros and cons of each one. But the fact is that there's different ones that are coming down the road, and we're excited about it. Now, what do you do about it? If you find the cancer by imaging, you have to ask yourself, okay, if it's really limited, 1 or 2 sites, maybe 3 or 4, can we do something about it? And if we can, should we do something about it? Okay? So here's a little bit of, let's look at biology and say, why should we do something about it? If somebody has a cancer that's already spread to a lymph node or to the bone or somewhere else, should we think about treating the prostate anyhow? Because one could argue the cat's out of the bag, should we do this? Well, here's one preclinical scientific study that shows that even when the cancer spreads to another site, it may be self-seeding. Those cancer cells that have spread to another site may then get back into the bloodstream and disseminate and still seed the prostate. They may change, they may become more aggressive even. So it gives a little bit of rationale for treating the primary lesion, for treating the prostate, even for somebody who's had the cancer spread. These slides are borrowed from Dr. Dan Lin, who made put together some real nice slides here, but this is another study that shows that even when it's spread to another site, that those other sites may also be seeding new sites. It's not all from the original prostate cancer. Once it's gone somewhere else, cells may be released from the other sites that may go on seeding. So not only might you think about treating the prostate, even in somebody who has advanced disease that is spread, you might think about treating those other sites that it is spread to as well. So this is a trial that's in planning. We actually have a trial coming here real soon that Dr. Bruce Montgomery is leading, but this is one through the largest national cooperative group in the country, SWOG, and Dr. Lin has been on the leadership committee to help lead this study. But it's the whole idea of saying if you have somebody that presents with limited metastatic disease, that means it's spread and maybe it's like 2 or 3 lesions, We'll enroll them and randomize them, which again, randomization means half get one treatment, half get the other treatment, to SST, which is standard systemic therapy, because the standard of care here is hormonal therapy, lowering the testosterone levels, okay, once it's spread. And now there's new data for adding chemotherapy that adds significant benefit. But you would get standard treatment on one arm, and the other arm would get standard treatment and then local therapy to all the sites that it is spread to. That means potentially surgery or radiation to the prostate, and potentially radiation to other sites that it might have spread to as well. And we'll follow the patients out to see if it improves time to the disease progresses, gets worse, or overall survival as well. So this is very exciting. It's a hot area in the field, is trying to find the cancer when it's limited spread and trying to either cure more people, and if we're not cure, at least lead to better survival outcomes by reducing, cytoreducing, again, that fancy word to reduce the tumor burden. Now the last section I want to talk about is, I told you we can use these imaging modalities to find the cancer, but can we use these imaging modalities not just to find the cancer, take a picture and tell us how aggressive the cancer is or not, but can we find the cancer there and treat it there? So there are some new modalities with antibody-based therapies that are bringing radiopharmaceuticals, or basically radiation that's being targeted to the cancer. This is the beta emitting radiation here, but this is an example of somebody who's received a few cycles of this to target towards PSMA and try to reduce the cancer. As you can see there, some of the spots are going away there as well, and this is gallium PSMA imaging. So it's pretty exciting. These are graphs of PSA change. Every single line represents a patient, and baseline where their PSA started. As you can see, most of the patients, the PSA went down. It didn't go up. Unfortunately, sometimes it does, but most of the patients responded to this modality of treatment. And just to finish up, there are other forms as well. There's a lot of things being developed. There's this alpha-emitting therapy as well, or actinium-225 PSMA. Now, some of you in the room may have heard of a drug called radium-223. This is a form of liquid radiation, as I call it. It's an alpha-emitting radiopharmaceutical. It goes to areas of the bone where it's uptaking radium, because it's on the same column of the periodic table as calcium. It goes into the bone. And the advantage to alpha-emitting radiopharmaceuticals is they don't penetrate deeply. They can only penetrate the thickness of a piece of paper. So it's kind of targeted therapy to one spot, because if it penetrates too far, it can lead to myelosuppression, which is lowering of your blood counts. White blood cells, red blood cells, they're all produced in the bone marrow there. So if you hit it too hard there, it spreads and leads to excess toxicity. You want to hit it right there where it's at and have very potent cell kill. So what they've done here is they fused that and targeted to PSMA, targeting to go to the sites that express PSMA. And the advantage is PSMA is not just in the bone. It's or when the prostate cancer goes to the bone, it's wherever it goes, the lymph nodes, the liver, wherever, and you can treat the cancer that way. Of course, there are potential downsides to it as well. It does go to the salivary gland, so there can be decreased saliva production. So there are pluses and minuses, but at least the efficacy, early outcomes, look pretty good in terms of PSA decline here with this. And again, this is right now not in the United States yet. It's only in Germany. We're working to bring it here, but the Lutetium PSMA, we're working with the group at Cornell that's doing a lot of this work, and we're hoping to bring the clinical trial here in the very near future to have that here as well. So I want to wrap up. Happy to take questions there in the question and answer section, but to summarize, and saying the whole point is can we detect earlier with these fancy imaging PET modalities, and can that help us aid treatment decision making? Can we treat those sites that it might have spread to early and lead to better outcomes that way? FACBC will be available very soon here. PSMA PET a little further down the road, but eventually we'll be doing research with this as well here. And then we have many oligometastatic disease trials again. Again, the idea is when it's spread just to a couple places, can we aggressively treat that with radiation and surgery, lead to better outcomes down the road? The future of these PET imaging modalities may go back to some of the stuff that I've done a lot of work on, is how can we measure response to therapy and intervene earlier for better outcomes. And we're really committed. The IPCR is absolutely dedicated to bringing all these things here. Thank you very much.